Welcome. Tonight's show is From Signals to Survival. It's going to be an overview of cellular communication. Everyone's had the experience where you get startled, scared, and your heart starts beating. That is all about a hormone that's called epinephrine, also known as adrenaline, and it's a huge part of the fight or flight response. The fight or flight response occurs throughout your entire body. You'll have decreased digestion, increased heart rate, your pupils will dilate, your bronchioles, those tubes in your lung, they'll dilate. And what we're going to focus on is what happens in your liver. What happens there is that epinephrine works to take a polysaccharide that you've learned about in Unit 1, glycogen, and convert it into glucose, a monosaccharide. So the question for tonight is, how did that happen? On the show tonight, we'll be talking about cell communication as our lesson of the week. We'll have a song of the week, my song, Cellular Communication. There'll be a weekly quiz, and there'll be tips for learning the greatest subject of all, which is biology. I am Glenn Wolkenfeld, also known as Mr. W. I'm a retired AP biology teacher, and my mission is to help you succeed in AP, IB, or college biology. I do that through this show. I also do that through my website, learn-biology.com, and my app called Biomania. I'm going to tell you more about those later. Before we get deeply into the show, I want to let you know that I've put together a checklist. It's called the Ultimate AP Bio Study Checklist. It'll tell you what to study when you're getting ready for that test on cell communication or when you're studying for the AP Bio exam. You download that by going to apbiosuccess.com/checklist. Let's start. Cell signaling. We're going to start with the big picture. Cells are constantly in communication with one another, and that communication takes two basic forms. There's direct cell-to-cell communication, and there's also communication via signals. So those signals are also called ligands or ligands. Those signals might be classified as hormones. That's when they travel a long distance from one part of the body to another part of the body. Or there are local regulators that move short distances. In plant cells, there are junctions between cells that are called plasmodesmata. These are bridges involving the cell wall and the membrane, and they allow signaling molecules, along with water and other substances, to diffuse from cell to cell. That's how plant cells can send messages from one cell to another. In animal cells, there are structures that are called gap junctions, and those gap junctions are built from protein channels. Those gap junctions allow substances, including signaling molecules, ligands, to move from the cytoplasm of one cell to the next. And that enables this cell to induce a change in this cell, because that's what communication is all about. It's about transferring information. It's about creating changes. Ligands are signaling molecules. What they do is bind with receptors in a target cell and have some kind of effect. Many ligands are hormones, things like estrogen, testosterone, adrenaline, growth hormone. What ligands do is they bind with receptors based on complementary shape. The binding leads to some kind of cellular response, some kind of effect. The binding between ligands and receptors is specific. It's as specific as that connection between enzymes and substrates that you learned about in Unit 3. And you know that every enzyme has a unique substrate that it binds with because it fits with it. It's the same thing with ligands and their receptors. I represented this ligand over here as a kind of circle binding with this part of the receptor. Here, I'm using a little bit more geometry to show this. There's a circle that binds with this complementary shaped receptor. And here's a triangle which binds with this complementary shaped receptor. But you know that biology is based on molecules like proteins, which have extraordinarily complex shapes. So a more realistic depiction is something like this, where a ligand that looks like this is binding with the receptor. The first form of cellular communication that we're going to talk about is quorum sensing. And quorum sensing is represented by these two images over here. The basic idea of quorum sensing is that bacteria change their behavior based on their density. And where we see this is in the formation of biofilms. Biofilms are these films of polysaccharide that are dense with bacteria that build up in areas where bacteria are living. Biofilms are what form plaque on teeth, and plaque on teeth are what lead to dental cavities. And the reason why we do things like floss and brush our teeth is to get that biofilm off of our teeth. Now let's talk about quorum sensing. The basic idea is that the biofilm won't form when the bacteria are in low density, but they'll form abundantly when the bacteria are in high density. How are the bacteria able to do that? This is a representation of the bacterial cell. And what the bacteria do is they're constantly releasing signaling molecules that bind to cytoplasmic receptors. Here's one over here at number 3. Here's another cytoplasmic receptor at number 5. When the bacteria are in low density, there just won't be that many ligands around. But when there are a lot of bacteria, then all of these receptors will start to be bound by the ligand. That's what we see on the right side of this diagram. What that will do is it will enable this receptor ligand complex to interact with the bacterial cell's DNA. That's what we see right over here. What that will do is it'll induce gene expression. So over here, we see something that looks like a polysaccharide at number 6. It's only produced when there's a lot of bacteria around. And the basic idea is that now we see a mechanism by which bacterial cells can communicate with one another through the release of a ligand binding with a receptor that causes genes to be turned on. It's just a very basic example of cell communication, just to emphasize that all cells communicate. Cell signaling involves 3 key phases, and this is stuff that you absolutely have to know. The first phase is reception of a ligand by a receptor. The second phase is signal transduction. Now, signal transduction means that that signal is going to be passed from the membrane deep into the cytoplasm. It's as if somebody came to the door and asked for somebody who was inside your house. They weren't allowed to get inside, and so you go to the door and you say, "Oh, who are you here for?" And they say, "Oh, I'm here to talk to somebody." And you say, "Hey, someone's here to talk to you." That's transduction. That leads to a cellular response. That's the basic 3-part model of cell communication: reception, transduction, response. And we'll be coming back to this diagram throughout. Now let's break down each of those phases. We'll start with reception. The signal molecule is going to bind with the receptor, and that receptor is embedded in the membrane. And as we said before, the binding is based on complementary shape. Now we're going to move on to the second phase, transduction, and the third phase, response. What will happen is that the receptor interacts with membrane proteins to produce a second messenger. It's represented by this oval over here. That brings the message into the cytoplasm, and 2 things can happen. That cytoplasm is filled with enzymes, and there might be some kind of communication process that turns those enzymes from dormant to active, from off to on. Another response is that this signaling system will make it into the nucleus, and then it'll activate genes. Everything that I've talked about so far relates to polar hormones. That's because those can't pass through the plasma membrane, which is made of phospholipids. So they have to bind at a receptor. That's how polar hormones work. That is very different from steroid hormones. What's an example of a steroid hormone? Testosterone, the masculinizing hormone, or estrogen, the feminizing hormone. Those are non-polar. They can diffuse right through the membrane. And what they'll do is they'll bind with a cytoplasmic receptor. Those cytoplasmic receptors will then form a receptor hormone complex. They'll diffuse through the nuclear membrane. They'll make it through a nuclear pore where they'll act as a transcription factor, and that'll lead genes to be transcribed into RNA. That RNA will make it out into the cytoplasm, and that will be then translated into protein. So steroid hormones make it into the cytoplasm, then they bind with the receptors, wind up activating genes, and that leads to long-term changes. Your success in AP Biology starts here. Are you struggling with AP Bio? With learn-biology.com, students get the skills and confidence to be a top student and earn 4s and 5s on the AP Bio exam, guaranteed. Go to learn-biology.com to find out how you can master your biology course and crush the AP Bio exam. What we're going to do now is we're going to do an illustrative example of cell signaling. We're going to talk about epinephrine and G protein coupled receptor systems. The context for this is what we talked about at the top of the show. It's the fight or flight response. You get scared, you get startled, you get psyched up, and you have this effect that goes all throughout your body. That response is largely related to a hormone that's called epinephrine. It's a key hormone in the fight or flight response. Here's epinephrine. This is the skeletal structural formula over here. And you can see this hydroxyl group over here, this hydroxyl group, this hydroxyl group, this amino group. It is polar. It's not going to make it through the cell membrane. So epinephrine, also known as adrenaline, it's polar, water soluble. It's released from the adrenal glands. The adrenal glands sit above your kidneys. Epinephrine's effects are widespread but tissue specific. So as I said earlier, epinephrine goes into the bloodstream from the adrenal glands. It goes everywhere. Some cells don't have receptors for adrenaline, for epinephrine. They don't do anything. Some cells do have receptors, and then what those cells do is based on the kind of tissue that they are. So in the stomach, epinephrine will cause digestion to decrease. In the heart, it'll cause the heart rate to increase. It'll cause your pupils to dilate. It'll cause your bronchioles to expand. We're going to focus on is this over here. In your liver, it's going to cause glycogen to be broken down into glucose. And remember from Unit 1, glycogen's a polysaccharide. It's made of glucose monomers. So what you've got to do is you've got to break all these bonds between the glucose monomers so they can be released into the bloodstream. Why do they have to be released into the bloodstream? That's all about Unit 3 of AP Biology, cellular respiration, so that glucose can power cellular respiration and make ATP. How does epinephrine induce the glycogen to glucose conversion? That's what we're going to focus on tonight. The way that epinephrine induces this glycogen to glucose conversion is through a G protein coupled receptor system that induces a phosphorylation cascade. G protein coupled receptor systems in liver cells look like this. Here's the liver. Here are some cells represented by this hexagonal shape. And here's the membrane of those liver cells. Now you can see the phospholipid bilayer. Here's the resting state when there's not epinephrine. The receptor is not bound by the ligand. There's no epinephrine in the system right now. There's a G protein nearby. Why is it called a G protein? It's called a G protein because of the way it interacts with GTP and GDP. Note that we have the system where we have a receptor, a G protein, and a nearby membrane-embedded enzyme that's called adenylyl cyclase. So no hormone. This is in its inactive off state. This is in its inactive off state. And therefore, the second messenger that's going to turn this system on is not activated. That's when the system is off. Now, you have this scary thing happen to you and you release epinephrine. Epinephrine binds at this binding site on the receptor, and it induces a change on the other side of the receptor. The receptor changes shape on its cytoplasmic side. The nearby G protein is still dormant. When it's dormant, it's bound to GDP. GDP stands for guanine diphosphate. And you've already learned about ADP and ATP, where ADP is the low energy form and ATP is the high energy form. It's exactly parallel. Let's look at this receptor in a closer way. Here's the ligand binding site, binds with epinephrine. There are 7 alpha helices. You learned about alpha helices back in Unit 1 when you learned about protein structure. And then there's this part over here that extends into the cytoplasm. And the thing to know is that when something binds over here, the binding can induce a ripple effect that ripples through these alpha helices and causes a change over here. So after epinephrine binds, it induces a change over here, and that activates the G protein. The G protein is randomly moving in the membrane. It'll bump into this part of the receptor, and that will induce a change in the G protein. That'll cause the G protein to let go of GDP and bind GTP. The G protein is now activated. That activated G protein bound to GTP drifts in the membrane until it bumps into adenylyl cyclase. There's a complementary fit between the G protein and adenylyl cyclase, and when the two bind, adenylyl cyclase, which was an inactive enzyme, becomes an active enzyme, and it will grab an ATP in the cytoplasm and convert it into cyclic AMP. Cyclic AMP is the second messenger. Here's a closer look. Here's ATP. Here's adenylyl cyclase. I didn't show it in the membrane. I'm just showing it in the typical iconic enzyme form. And it takes ATP, chops off 2 phosphates, and creates cyclic AMP. It's called cyclic AMP because of the way that the phosphate is bound to the adenine. What do you need to know? Cyclic AMP is a modified form of ATP. It's the second messenger. What we're going to do now is talk about transduction and amplification. Cyclic AMP, which is the second messenger, is going to activate a chain of relay molecules. Those relay molecules are called kinases or kinases. That activation involves something called a phosphorylation cascade. A phosphorylation cascade involves adding phosphates, adding phosphates, adding phosphates. How does that work? Kinases are these relay molecules, and they're proteins that are activated when they're phosphorylated. So on the left side, we have unphosphorylated kinases. And what we see over here is that because of the presence of cyclic AMP, this kinase over here becomes an activated kinase over here. And once it's activated, it'll activate the next kinase in the chain. Now, there's a couple of other things that I couldn't really convey in this diagram, but there's a level of specificity. In other words, this protein kinase 1 will only activate protein kinase 2. It'll grab a phosphate and it'll glom it onto protein kinase 2, which will activate protein kinase 2. And then protein kinase 2 will activate protein kinase 3. Protein kinase 3 will become activated. And this will go on in a long chain. And then at the end, we'll get to the terminal enzyme. The terminal enzyme, I'm just representing by the letters GP, but that stands for glycogen phosphorylase. And what that does is that's the enzyme that takes glycogen and snips its glucose subunits off to release glucose into the bloodstream. What I want you to start imagining now is that there are multiple phosphorylation cascades that happen in parallel with one another. Each step involves multiple activations, and the result is signal amplification, where one epinephrine binds, but that'll lead to the production of several cyclic AMPs by adenylyl cyclase. Each cyclic AMP will kick off protein kinase 1, and then you have to imagine a whole chain, and that'll activate so many glycogen phosphorylases. We've amplified the system. And the way to think about this is that your liver cells are ready to do this. They're ready to take their glycogen and instantly release it into the bloodstream so that it can support the body in the fight or flight response. But they don't do it until the signal comes in. Once the signal comes in, those enzymes spring into action. They release glucose after breaking down glycogen into glucose. We finally get to the terminal enzyme, glycogen phosphorylase. And what that does is it takes glycogen and snips all these glucoses off so that the glucoses get released into the bloodstream. And there we have our response. So think about the ground that we've covered. We started with reception. We've gone through transduction. release of cyclic AMP. And then finally, we saw a phosphorylation cascade that led to a response. How do we turn this thing off? So the end of the thread leads to the end of epinephrine secretion. Why? Because the brain will stop sending a signal to the adrenals and say, no more need for epinephrine secretion. When that happens, the epinephrine is going to diffuse away from the receptor. That epinephrine was never tightly bound to the receptor. It fit there because it had a complementary shape. Maybe there was some hydrogen bonding, and something that's only connected by a hydrogen bond is not going to stick there forever. So epinephrine is no longer secreted, and the epinephrine is going to diffuse away from the receptor. That is going to cause a change in the receptor shape over here. And at a certain point, the G protein will take its GTP, break off a phosphate, which will render it into GDP. So now the G protein is turned off. Now that the G protein is turned off, adenyl cyclase is going to deactivate. That is going to stop the conversion of ATP to cyclic AMP. So we've got no more second messenger. So that's the beginning of the shutdown of this system. The thread is gone. We're not going to produce any more cyclic AMP. But then there's also things that happen in the cytoplasm. There are enzymes that are called protein phosphatases. And what they do is they take these phosphorylated kinases and they dephosphorylate them. They break their phosphate off and render them back into their inactive state. So again, you have to remember that your cells are like a football team that is ready to go, waiting for the quarterback to say, hut, go. And when they do say go, then you have these phosphorylation cascades. But then the play ends and everybody goes back to the inactive waiting state. So the cell is capable of instantly turning on and turning off. And then the cytoplasmic signal transmission ends, and we're not going to make any more glucose from glycogen. So folks, I have a song that I've created about cellular communication, and it summarizes a lot of what we've learned, and I'm going to play it for you right now. I'm going to go like this. Have you ever had a sudden fright? Perceived a threat, you're all geared up for fight or flight. Heart pounding, face flushed, hair standing up on end. It's partly from a hormone called epinephrine. Epinephrine's got another name, adrenaline. Secreted from adrenal glands, it's stuff in EpiPens, a shot can save your life. If you have an allergy involving sensitivity to nuts or stink from bees, epinephrine's polar so it can't diffuse through the lipid bilayer. So instead, it induces a second messenger, it's cyclic AMP, which stimulates massive cellular activity. Let's learn how liver cells respond to an epinephrine dose. What they do is break down glycogen to glucose, which streams into your blood to give you energy required to respond to threats as you fight or flee. How does epinephrine get your liver cell to do this function? It's 3 steps. First, reception and transduction, then cellular response. In enzyme activation, we're learning about the process of cell communication. Cellular communication works through phases 3. Reception of a ligand, they're complementary. Second messenger release, transduction is a beast. No amplified response until the stimulus has ceased. Today's focus is G protein coupled receptors. We'll leave the other systems for another lecture. Epinephrine's the ligand that with the receptor binds. It's a hormone, the hydrophilic kind. Ligands bind to receptors only if the shape matches its specific, based on complementary attachment. Epinephrine goes everywhere but only affects cells with receptors with which it can connect. Once epinephrine binds at the receptor binding site, the receptor changes shape on its cytoplasmic side. Or just inside the membrane you'll find the G protein, which until epinephrine binds is dormant, asleep in its dormant form. The G protein's bound to GDP, but epinephrine binding makes it bind with GTP. Then the G protein drifts in the membrane space till it bumps into adenyl cyclase, a membrane-bound enzyme whose signaling functions activating a second messenger, setting up transduction. Transduction converts a message into another kind. Message 1 was a ligand, which came along to bind with the receptor, which in response woke up the G protein, which gets adenyl cyclase to take an ATP and remove 2 phosphates, making cyclic AMP. That's the second messenger. It takes a message deep into the cytoplasm, where it unleashes a chain of enzyme action, a phosphorylation cascade. The message was received, it was then transduced. Next we'll see how cellular response gets produced. Cellular communication works through phases 3. Reception of a ligand, complementary. Second messenger released. Transduction is a beast. No amplified response until the stimulus has ceased. A phosphorylation cascade involves a chain of enzymes known as kinases. These kinases get phosphorylated, which means gaining a phosphate, which activates them. And what do kinases do when they're charged up with a phosphate? They find the chain's next kinase and phosphorylate it. This chain of activation is like falling dominoes, because that's how a phosphorylation cascade goes. And each kinase carries out multiple activations, causing massive signal amplification. The signal grows in strength with every kinase action, building in intensity, a chain reaction. One epinephrine binds, causing many cyclic AMPs, each of which activates many kinase proteins. By the chain's last kinase, tens of thousands activated. A cellular tsunami as the chain is terminated. Glycogen phosphorylase is the enzyme at the end that makes glucose by hydrolyzing glycogen. So into the blood from the liver, glucose gets secreted, and the threat that started the story can be fled from or defeated. It's adaptive how your cells are poised to spring into action. Once they hear epinephrine's alarm, there's a reaction in all eukaryotes, from amoebas to acacias. G-protein coupled receptors used in cell communication. Cellular communication works through phases 3. Reception of the ligand, they're complementary. Second messenger released. Transduction is a beast. No amplified response until the stimulus has ceased. Back to the ligand of the receptor. It only stays for a moment before it diffuses away. So when the thread is gone, the cascade gets shut down. With no adrenal secretion, the receptor's unbound. G-protein drops the phosphate, now bound to GDP. It goes back to sleep, stops its activity. Bound to GDP, it no longer stimulates adenylyl cyclase, which no longer creates cyclic AMP, so the second message stops. Kinase phosphorylation quickly drops as other enzymes, protein phosphatases, clip off phosphates, turn off kinases. Glycogen phosphorylase stops hydrolyzing glycogen, so blood glucose normalizes. Liver cells return to their resting state. I love how G proteins let cells communicate. Let me bring up the weekly quiz, which is going to be super fun. Every unit in AP Biology on learn-biology.com has a unit review page. We're just going to do a couple of multiple choice questions. Plasmodesmata, shown below, are channels through which plants can send chemical signals between one cell and the next. Researchers are testing the effects of a drug that inhibits the formation of plasmodesmata on plant growth. They tested 2 concentrations of this drug on the average growth of the plant in 5 trials. Here's a table that's showing what happens after 5 days, 10 days, and 20 days. Which of the following is the best explanation for the effect of the drug? I'm going to explain what's going on in the table. But 5 days in the control group, which didn't get any of the drug, there was 2 centimeters plus or minus 0.5 of growth. In the group that had 10 microliters of the drug, there was also 2 centimeters plus or minus 7. And then with 20 microliters of the drug, it was 1 centimeter plus or minus 0.5. That's after 5 days. After 10 days, the control had grown 5 centimeters, the 10 microliter group 3, and the 20 microliter group 1. At 20 days, the control had grown 10, 10 microliter had grown 5 centimeters, and the 20 microliter had grown just 1 centimeter. So, which of the following is the best explanation for the effect of the drug? 1, the drug makes it more difficult for molecules that stimulate cell division to move from cell to cell, inhibiting plant growth. 2, the drug makes it easier for molecules that stimulate cell division to move from cell to cell, stimulating plant growth. 3, this drug makes it easier for molecules that inhibit the cell cycle to move from cell to cell, inhibiting plant growth. 4, this drug makes it more difficult for molecules that inhibit the cell cycle to move from cell to cell, inhibiting plant growth. So, folks, what do we got? Let's go with number 1. The drug makes it more difficult for molecules that stimulate cell division to move from cell to cell, inhibiting plant growth. Way to go. Without plasmodesmata, signaling molecules would be prevented from moving from one cell to the next. If these molecules stimulated cell division, then plant growth would be inhibited. Second question. Which of the following mechanisms of cell communication are not found in animal cells? 1, long-distance signaling through hormones that bind to receptors in target cells. That was choice 1. Choice 2, direct cell-to-cell contact. Choice 3, transfer of signaling molecules via plasmodesmata. Choice 4, short distance signaling through the release of ligands that bind to nearby receptors. Which of these is not, I repeat, not found in animal cells? I see a bunch of threes. Holy cow, we got a bunch of superheroes in the crowd. Indeed, plasmodesmata are found in plant cells, not animal cells. During many cell signaling processes, the enzyme adenylyl cyclase converts the molecule below into an important second messenger. As part of this conversion, which bond is broken? Is it the bond at A, B, C, D, or E? Okay, let's choose A. No. Let me tell you why. Remember that the signaling molecule is cyclic AMP. That A stands for monophosphate, one phosphate. So if you break this bond, you'd still have two phosphates. If you break this bond, you'd be down to one phosphate. And then if you hooked this part to the sugar over here, then you'd have AMP. So the answer was B, not A. But this is the beauty of learned biology. If you get it wrong, you get another try. Epinephrine, also known as adrenaline, is produced by the adrenal glands. The structural formula for epinephrine is below. Diagrams S and T below are general models for signal reception and transduction. Here's model S. Here's model T. Which of the following correctly identifies and explains the model that corresponds to how epinephrine works? Choice 1, model S, because it shows epinephrine diffusing through the cell membrane and activating genes. Choice 2, model S, because it shows epinephrine binding with a cytoplasmic receptor. Choice 3, model T, because it shows epinephrine phosphorylating a relay molecule. Choice 4, model T, because it shows epinephrine binding with a membrane receptor. What do we got? We have 1, 2, 3, or 4. Think about what we just learned. Epinephrine is polar, water soluble. Water soluble things can't diffuse through the phospholipid bilayer. So that eliminates model S. So choice 1 and choice 2 are out. This third choice, model T, because it shows epinephrine phosphorylating a relay molecule, the epinephrine binds over here. It doesn't phosphorylate the relay molecule. So really what we're down to is model T, because it shows epinephrine binding with a membrane receptor. Your plan for this week is go to learn-biology.com, then courses, AP Bio, Unit 4, and then you would do these tutorials that would take you through much of the territory that I covered tonight, but it would take you through it better because you'd interact with it and you would learn the material super well. That is what it's all about. You got to interact with biology to learn it. That's what learn-biology.com is all about. I'll have another video coming up soon. Thank you so much.